Thank you, Julian, for the introduction. It's a pleasure to be here tonight. I've only just realized that there is a spelling mistake in my surname in the poster, but that's okay. Probably nobody makes it after the Cerci, so it's okay. This image that might look rather unimpressive to you, it's actually the very first MR image which was published in 1973 by Paul Lauterbur. And it represents the image of two vials containing liquid. And the image was obtained by rotating the object and taking projections along different planes. This technique was named by Paul Lauterbur NMR zeugmatography because it exploited the combination of a static magnetic field and the radiofrequency magnetic field. And the Greek word zeugma means what joins, and the concept was that this image was represented the coupling between these two magnetic fields. It is also important to mention that Paul Lauterbur shared with Peter Mansfield the Nobel Prize in 2003 for the introduction of MRI. And this is not just due to the fact that he realized these images, but also to the fact that he was actually able to foresee the great potential of this technique. And in fact, this is what it says in his Nature paper. The variations in water contents and proton relaxation times among biological tissues should permit the generation of useful zeugmatographic images, selectively picturing the various soft structures and tissues. And he was definitely right if we compare this image published in 1973 with the type of images that are actually achievable 40 years later. This is an image of the brain of really high quality obtained at a 7 Tesla scanner, and we'll see in a minute what that means. But the reason why I put these two images next to each other is to highlight the fact that even 30 years' time there's been such a great evolution. It's also down to the work of MRI physicists. So first of all, for those of you who are not completely familiar with the MRI, I just wanted to give a little bit of background. And I thought that it would be a good point to start to point out the differences between MRI and other types of radiological techniques that are also used to image the brain. So this image that you see here is a CT scan of the brain. And clearly, you can use the CT to image the brain. But if you compare it to MRI, the properties of these two techniques are quite different. And what makes MRI particularly attractive is, first of all, the fact that it does not imply the use of ionizing radiation, which makes it very safe. So people can actually have several MRI scans without any associated risk. It can acquire images in different scan planes, and these are some examples. And it gives excellent detail of soft tissue. This can already be perceived from this image, which is of not particularly high quality, but this is the type of images that you can obtain when you push things a little bit further. So these are images of a deep structure of the brain stem, which is of about 1 centimeter square in area, and these images are obtained at the 7 Tesla scanner installed in Pisa, which is a rather advanced research facility. But what makes MRI really different is the fact that it can produce images with a very wide range of contrast. So not only one type of image, but several types of images. And this is also what makes it ideal for developing quantitative methods. So these three images represent really the three basic types of contrast that you can obtain with MRI. The three basic types used in routine clinical imaging, which are called proton density weighted, T2 weighted, and T1 weighted. And these three images already can be sensitive to different properties of tissue. But we will see today that MRI goes a long way beyond this. So what do you need to do MRI? From a hardware point of view, you need obviously a magnet, as it's summarized by the word magnetic. You need an RF coil to produce a radiofrequency field, which needs to be tuned at a specific frequency, and this can be linked to the word resonance. And you need a gradient coil, which is used to encode where the signal comes from in your patient, and this obviously can be linked to the word imaging. But in order to obtain images, you also need to, oh no, sorry, I just want to mention that for people who are not familiar with the magnetic fields, that I've already mentioned the word Tesla previously. That's the unit measure of magnetic fields. And just to give you an idea of the entity of magnets that we work with in MRI, the Earth magnetic field measures approximately 6 times to 10 to the minus 5 Tesla, which means 0.00006 Tesla. A fridge magnet is of approximately 5 times 10 to the minus 3 Tesla, so that's 0.005. And typically, clinical MRI scanners operate at 1.5 Tesla. So this is already a huge magnetic field compared to a fridge magnet. But nowadays, human scanners can operate at something like 7 Tesla or 9.4 Tesla. The MRI signal comes from the water that is in the human body. So you know that we're made up of approximately 60% of water. And the interesting property of water, the water molecule, is that it contains 2 hydrogen atoms, and the hydrogen nucleus has several physical properties that make it interesting. But the one that is relevant for MRI is called the magnetic moment or spin. And this basically makes the hydrogen nucleus behave like a tiny magnet that produces its own magnetic field. Now, this might sound surprising because in our everyday experience, we don't appreciate the magnetic properties of hydrogen contained in water. But this is due to the fact that in normal conditions, every little magnet associated with the hydrogen nucleus is oriented randomly, and therefore these effects cancel each other out. But if you think about what happens, for example, to a compass needle when you put it inside a magnetic field, it will tend to align with the north pole pointing towards the magnetic field itself. And something similar happens with the hydrogen nuclei, although these effects are slightly more complicated. Just to summarize briefly, what happens is that all the magnetic moments of the nuclei will start to precess, which means that they will rotate a little bit like a spinning gyroscope that precesses about the gravitational field of the Earth. And when placed in the magnetic field, we will have all the nuclei tending to align, either pointing along the magnetic field or in opposite direction to the magnetic field. And we will have approximately half of them pointing upwards and half of them pointing downwards, but there is a tiny difference between these 2 populations that macroscopically produces a global magnetization. And it's by manipulating this magnetization through the use of radio frequency magnetic fields and gradients, magnetic field gradients, that we're able to produce images using MRI. So MRI really is great for spotting abnormalities in several organs, but in particular in the brain. And this is because it's extremely sensitive to pathologies. For example, if I show you these images, even if you're not an expert of the brain, it's fairly easy to spot that there is something that shouldn't be there. And similarly, if I showed you an image like this, so obviously this has a very important potential in terms of diagnosis. And indeed, these 2 scans belong to a patient with multiple sclerosis and the patient with stroke. But one limitation of conventional imaging is that sometimes different diseases can look very similar. For example, if I showed you this other scan, which appears at your right-hand side at the bottom, it doesn't look very different from the multiple sclerosis scan. Or in some other cases, brain scans may look almost normal, like this, but belong to patients who have actually a clinical presentation that suggests otherwise. And indeed, the patient on the right corner, the bottom corner, presents with early cognitive decline, so quite a different clinical presentation compared to the multiple sclerosis patient, while the guy at the top has Alzheimer's disease, so comparatively much worse clinical condition than the patient underneath. So what we want to do with quantitative MRI is try to go a little bit further in understanding what is going on in tissues. And to see this a little clearer, I'm going to talk to you a little bit, for example, about the applications in multiple sclerosis. So multiple sclerosis is a chronic inflammatory disease of the central nervous system, and it's a major cause of neurological disability in young adults in Western world populations. In the majority of cases, it presents in the so-called relapsing remitting form, which is characterized by acute episodes that tend to resolve themselves in time and alternate with periods of remission. After the attacks, patients tend to recover completely or partially. Unfortunately, after a variable period of time, most patients convert to the so-called secondary progressive form, where the acute episodes tend to disappear, but a progressive accumulation of disability starts. It's a disease of the central nervous system, so it affects the cells in our brain, which are the neurons. So this is a schematic representation of the neuron, which is characterized by the cell body, or soma, which is the part that fires the neuronal signal, and the axon, which is the part that is responsible for delivering the signal. So it's a little bit like an electrical wire, and for this reason, the axon is wrapped by this insulating material, which is called myelin, and enables the signal to be transmitted fast and efficiently. So in the brain, typically the cell body is located in the gray matter, and the axon is located in the white matter. In multiple sclerosis, what happens is that the immune system, for reasons that are still not completely understood, instead of attacking external agents that come in contact with our body, started to attack the myelin instead. And this causes scar that tend to slow down the neuronal signal transmission. This is at least what happens at the initial phases of the disease. Radiologically, MS typically presents with multifocal white matter lesions that can be seen very clearly on T2-weighted scans. However, there is so MRI is extremely sensitive to the pathology of multiple sclerosis, so to the point that nowadays it is possible to diagnose multiple sclerosis based solely on MRI criteria. However, there is also a well-known mismatch between the radiological aspects of multiple sclerosis and the clinical aspects of multiple sclerosis. In fact, the scan on the right-hand side also belongs to a patient with a diagnosis of MS, but you can see that the number and extent of lesions in this patient is much less than in this one, and despite this, the patient on the left-hand side clinically is actually doing much better than the patient on the right-hand side. The expanded disability status scale is the most widely used scale to measure disability in MS and runs from 0 to 10, where 1 basically means no disability. So there are several explanations for this paradox, but one of the things to understand is that, first of all, even though lesions all look the same on T2-weighted scans, they might actually correspond to very different pathological substrates. And additionally, we know that now that despite this part of the brain in this patient might look normal on a T2-weighted scan, we know that it's not actually normal, to the point that the term normal appearing white matter has been introduced to describe this tissue. And also, despite being considered traditionally a disease of the white matter only, we now know that the gray matter is involved from the very beginning of the disease. And therefore, the damage to the gray matter might also have an important impact on the clinical evolution of the disease. So what we really want to do is to try and go beyond the look of these images, which can be of great support in the diagnostic phase. But what we want to do is go beyond the resolution of this image and try to see what is happening underneath and try to look at the microstructure of tissue underneath lesions and underneath the normal appearing tissue. And this is where quantitative MRI or microstructural imaging can be of help. So when I teach the medical students, I use this example to try and explain more clearly the difference between conventional and quantitative MRI. So I asked them to explain to me in conventional MRI terms what the difference between these two pencils are. And so in qualitative terms, you would reply, B is shorter than A. So what we want to do with the quantitative MRI is actually do a quantitative measure of this difference. So take a ruler, look at the length of pencil A and then at the length of pencil B, and take the difference, and so produce a number that tells us something that can be compared between different populations and different patients, hopefully. So my personal journey through the world of MRI started in the summer of 1997 when I was about to graduate in telecommunication engineering. And I heard that the imaging center located within the San Raffaele Hospital in Milan were looking for a young person graduated in a numerate subject that they wanted to form to become an MRI physicist. So at that point, I was left with the dilemma to either go and took a standard engineering job where I would be sitting with hundreds of nerdy engineers, or go and work with medical doctors. So at this point, obviously my choice was fairly easy, except that I soon found out that they don't all look like that. But more seriously, what attracted me to this job was the fact that it would give me the opportunity to work in a multidisciplinary environment. And also, this might surprise some of you, the fact that they promised me that if I took the job, they would send me to England to learn everything I needed to know about MRI. So in January 1998, I packed my things and moved to Leicester in the Midlands, where I ended up working with Mark Horsfield, who was my first mentor, and certainly a person I owe quite a lot in terms of the development of my career. So I went to Leicester to learn about a technique that was just exploding in those years, which is called diffusion-weighted imaging. So diffusion is a physical process that probably most of you have come across in their undergraduate, well, actually in their high school physics lectures. It's well represented by this movie, although when I talk about it, I prefer to mention wine being poured very slowly in a glass of water. Well, this yellow liquid certainly doesn't look like wine. But typically, when it refers to the mixing of two liquids, except that when we talk about, and it's due to the fact that the second liquid particles slowly move around the water that contains it and to reach a complete mixing up. But when we talk about diffusion in the brain, it's a slightly different concept, because in this case we're looking at water molecules diffusing within water itself. So sometimes we talk about self-diffusion in this case. So diffusion can be measured, I would say, relatively easy with MRI, and the reason for this is that MRI is naturally sensitive to motion, as a lot of people would have experienced when patients are not very compliant. So because the MRI is sensitive to any type of motion, it's also sensitive to microscopic motion, such as diffusion. And the reason why it's useful to measure diffusion in biological tissues and in the brain in particular is that when water molecules diffuse, they will encounter obstacles which are constituted by the organelles and membranes that constitute the tissue itself. And so the presence of these obstacles will actually influence the motion of the water molecules. So if we can measure the motion of the water molecules, we can indirectly infer about the microstructure of tissue. So this image here shows more or less what happens in the gray matter of the brain, where we have the cell body of neurons. And because these cells are not distributed with a specific alignment, the water molecules tend to move randomly in all directions with more or less the same probability. What happens in the white matter is quite different, because in this case, the white matter fibers are organized in bundles of fibers, and therefore the water molecules have much higher probability of moving along the axon direction than across it. In the first case, we say the diffusion motion to be isotropic, while in the second case, we say it to be anisotropic, then both words come from Greek. So because with MRI we can really measure diffusion only along one direction at the time, we are posed with the problem that if we put a patient inside the scanner and we take two different pictures with the diffusion gradients along different directions, which therefore measure diffusion along two different directions, we're going to end up with images that look quite different. So these are images taken exactly during the same scan from the same healthy volunteers. And these hyperintensities that you see here have got nothing to do with pathology, but simply reflect the areas of the brain where the alignment of the white matter is perpendicular to the diffusion of to the direction of the diffusion gradients. And this slide just reiterated the concept that diffusion has this property in the white matter because the motion along the direction of the axon is much faster than in direction across it. So in those years when I moved to Leicester, this technique, diffusion-weighted imaging, was just becoming very popular at the time. And so in order to face this problem of diffusion anisotropy, there's basically two strategies that you can pursue. One is to try and get rid of this anisotropy problem, and so try to obtain measures that are averaged by directionality. And so the first task that I was given was to decide to design an acquisition sequence with these properties, which basically allowed to acquire very quickly images that were not biased by this direction problem. And this is an example of a scan of a patient with multiple ischemic lesions. You can see them appearing as hyperintensities on the T2-weighted scan. And diffusion imaging has the property of being able to distinguish between acute and chronic ischemic lesions. In fact, I don't know if you can appreciate that one of these lesions appears dark on the diffusion coefficient map, while the others appear bright, just like they do in the T2-weighted scan. So this has become now the standard approach to measure, to image acute strokes, diffusion-weighted imaging. The second approach that you can take in order to take anisotropy into account is to use this so-called diffusion tensor imaging, which was introduced by Peter Busser in 1998. So without going into too much detail or what a tensor is, it's a mathematical entity which is often used to describe anisotropic phenomena in physics. And from a geometrical point of view, it can represented by something called the ellipsoid, which has more or less the shape of a rugby ball. So no jokes are permitted about England versus Italy game. So using this approach, which requires a slightly more complicated image acquisition and image analysis, we're actually able not only to obtain images that are not dependent on the specific orientation of the patient within the magnet, but also to estimate the degree of anisotropy. And you can appreciate on the anisotropy map the contrast between white matter, where diffusion is anisotropic, and gray matter, where diffusion is isotropic, but also to extract the principal direction of diffusion, which appears to coincide with the expected direction of white matter tracks in that specific location, like in this case, which is the splenium of the corpus callosum, a structure that connects the two hemispheres of the brain. And this information can be also color-coded, obtaining these pretty-looking images where everything that is shown in red represents fibers running from left to right and vice versa, while everything that is in green represents fibers running from anterior to posterior direction or vice versa, and everything that is blue represents fibers running from superior to inferior or vice versa. And looking at this image, you can really start to see a lot going on in the white matter, while when looking at conventional MRI images, the white matter looks all pretty much the same. So this technique really has represented a big change in the way that people look at the white matter. So I did my job when I was in Leicester and developed all these methods that at the time were not available on commercial scanners. And then in December 1998, I made my way back to Milan, where I started to apply all these techniques, in particular to multiple sclerosis, which was the focus of the imaging center where I worked. So these images that nowadays would look pretty awful were quite desirable at the time. And this was one of the studies that we did then. And what I'd like to attract your attention to is the fact that on this conventional image here, you see the patient has got a lot of confluent lesions here that all look pretty much the same on the scan. But if you look at the diffusion coefficient map on the side, you can see that lesions that look identical on the proton density weighted scan look very different on the diffusion weighted map. So this tells us that the diffusion coefficient differs between different types of lesion, and this certainly corresponds to different type of pathology underneath. So, so at that time, we were able also to show other properties of tissue in multiple sclerosis using the diffusion coefficient, such as the fact that the diffusion coefficient is also altered in the normal appearing white matter of patients compared to the white matter of healthy controls. We did quite a lot of studies using diffusion in MS in those days because it was a technique that wasn't available to many centers, so we were one of those who could do this. And these are also some interesting data where we looked at the formation of new lesions in MS patients. So while on T2 weighted scans, all lesions tend to appear the same. If you inject your patient with a contrast medium called gadolinium, this is sensitive to the presence of inflammation and therefore tends to highlight new lesions, like in this particular case. So this is typically a new lesion. And so what we did was to scan a bunch of patients weekly for a long period and then go and identify all the new lesions using post-gadolinium scans, and went back to the previous scans and look at the diffusion coefficient in the normal appearing white matter corresponding to the area where patients were going to develop a lesion. We did this also in some control area that was not going to develop a lesion, and we observed that the diffusion coefficient was indeed remaining stable, while in the area that were going to develop a lesion later on, we could see an increase in the diffusion coefficient up to 6 weeks before the appearance of lesion. So this is telling us something about the complicated pathological processes that are going on in the normal appearing tissue before we can actually detect the lesion using conventional methods. So it was kind of easy to think about applying MRI techniques to multiple sclerosis because the radiological signs of multiple sclerosis are so clear. But when all these quantitative methods started to be available, it also became apparent that they had a great potential in diseases that with conventional imaging are not so easily characterized. And in particular, we started to look at Alzheimer's disease, which is a condition that in most cases presents with a memory deficit that tends to worsen rapidly, and that this worsening of symptoms is known to be related to the progressive involvement of different areas of the brain. We also know that there's quite a lot of microscopic changes that go on in the brain during normal aging. So these are all things that unfortunately are happening to all of us. But in particular, it's the increase of neuritic plaques and the increase in neurofibrillary tangles that occurs disproportionately in Alzheimer's disease. And this is really the pathological signature of Alzheimer's disease. From a conventional imaging point of view, really, there is no spatial features of Alzheimer's disease, with the exception of a generalized atrophy, but not in all cases, that is particularly marked in the mediotemporal lobe. Like this scan shows, you can see this hole here in this structure that is called the hippocampus, that you can clearly see in the healthy elderly control, and it's not, basically it's very atrophic in the patient. But this distribution of atrophy can be quite aspecific, and so therefore the role of conventional imaging in Alzheimer's disease really is just about excluding the presence of other pathologies that could explain the same symptoms. So it is particularly interesting to try and use quantitative MRI to characterize this type of diseases where conventional imaging doesn't really show much. And in those days, Alzheimer's disease was considered primarily a disease of the gray matter, so in a way, a complementary model to multiple sclerosis. But we started looking at the white matter, thinking that if the disease progresses and spreads through the brain, then it makes sense to imagine that the white matter must have some involvement. And indeed, we were able to show using diffusion imaging not only that the white matter is involved in the sense that there are pathological changes, but also that these changes correlate quite well with the global cognitive impairment in this patient. So it definitely shows that the damage to the white matter has a role in Alzheimer's disease. So in the summer of 2002, my friend Derek Jones, whom I'd met while I was in Leicester, he was working through his PhD, is now the head of the Kubrick, which is the imaging center at the University of Cardiff. He got married and invited me to his wedding. And because it was an MRI physicist wedding, it was full of MRI physicists from the UK. And in particular, Gareth Barker, who is sitting in the second row tonight, was there. And after a few drinks, he made the big mistake of talking to me about a job opportunity at UCL, at the Institute of Neurology. So in the November of 2002, I packed my things and again moved to the UK, this time to London, where I ended up working for Maria Ron, who was the head of neuropsychiatry at the time. So my job really was to implement quantitative MRI methods for the study of psychiatric disorders. But while I was there, I became interested in a different imaging technique, which is called magnetization transfer, which is based on quite a different principle than diffusion imaging. So from a practical point of view, what you observe when you do an MR experiment, so you can obtain a standard image like this one, a proton density image, and then if you add to your acquisition some special radio frequency pulses that are tuned at a frequency different from the one that you would normally use for an MR experiment, what you observe is that the contrast in the image changes, and in particular, you observe a signal attenuation that is more marked in some areas of the brain than in others. And this is due to this magnetization transfer effect that basically summarizes the interaction between the hydrogen proton in the water molecules that we've seen earlier produces the MR signal. Oops, sorry. And the the hydrogen that is present in other forms in biological tissues, and in particular in macromolecules such as lipids and proteins. So this second type of hydrogen protons do not directly contribute to the MR signal because basically we can't image them directly, but they interact somehow with the hydrogen in the water molecules, and therefore using these special radiofrequency pulses that I mentioned before, we can affect the macromolecular protons, and because of the exchange between the macromolecular protons and the water protons, we will observe indirectly an effect. For example, this can be appreciated by taking an egg and boiling it. So what happens when you boil an egg is that the proteins that are in the white tend to form a new type of links and form long chains of proteins that have magnetization transfer properties. So then if you put your egg in the scanner and take two different images, one where you use these special radiofrequency pulses and one where you don't, so with your naked eye, you don't really appreciate a big difference between these two images. But if you try to use a more quantitative approach, which is to take the percentage difference between the signal intensity in these two images, you will obtain a map that looks more or less like this. What is interesting about this, which is called the magnetization transfer ratio map, is that there is basically no signal in the yolk. And this is because you don't have these special type of proteins in this part of the egg. Well, you do have them in the white, and therefore they show an MTR effect. This is important because one of the most represented lipid proteins in our brain is myelin. And therefore, magnetization transfer is indeed sensitive to the presence of myelin, as can be appreciated by this example of a patient with multiple sclerosis. So this T2-weighted scan shows multiple lesions. And you can appreciate that the lesions appear darker on the MTR scan, just like the egg yolk appeared in the previous slide. And it has indeed been shown that the MTR does correlate with the amount of myelin measured in post-mortem brain. So magnetization transfer ratio has been around for quite a long time, but what happens when you do a magnetization transfer experiment is that you can use different types of radiofrequency pulses, and when you play a little bit with the characteristic of these pulses, what you see is that your signal will distribute along curves that look more or less like this. So if you just simply do an MTR experiment, what you're really doing is taking two images at two different points along this curve, and you will obtain images with different contrasts and then compute the percentage difference. So this is great. We know that it correlates with myelin to some extent. But if you look at this graph, you certainly notice that we're actually throwing away an awful lot of information. There is so much more in there, and it would be good to be able and exploit this a little bit more. So this is really what quantitative magnetization transfer imaging is about. And to make you understand a little bit more how it works, I have to introduce the concept of model fitting, which is an approach that in physics is used very often to describe a phenomenon. So you basically introduce a mathematical equation that you think can describe your phenomenon, and then this equation will depend on two different sets of parameters, some parameters that you can control experimentally and some parameters that you don't know and you want to estimate. So if the model is linear, which means that can be represented by a straight line, typically the parameters that you want to estimate are the slope or the intercept of your line. And things are slightly more complicated when the model is non-linear, but the concept is very similar. So with magnetization transfer, what you are doing is basically varying the type of magnetization transfer pulses that you use, and this will give you a series of measurements that distribute in the space of your experimental parameters like this. And then you fit your equation that has been worked out by clever people and estimate parameters that describe the magnetization transfer properties of the tissue. Among these parameters that you can extract from MT, it's of particular interest the relative size of the macromolecular pool, which is called F. And the reason why this F is interesting is because it basically represents the amount of macromolecular protons, hydrogen protons, compared to the water hydrogen protons. And because myelin is such a big contributor to the macromolecular protons in the brain, this parameter is actually believed to be a proxy of myelination. This is an F map, and again, you can see how the F is much higher in the white matter than in the gray matter. And this has been demonstrated by several animal work, and in particular, these are data that I've analyzed in collaboration with the Neurological Institute, BESTA, in a model of reversible demyelination and remyelination, which is cuprizone. Cuprizone is something that you can feed to mice, and because it kills the oligodendrocytes that produce the myelin in the brain, it will cause, after some weeks of treatment, the complete disappearance of myelin. This is an F map of the mice before treatment, where you can see this hyperintense structure represented, the corpus callosum, which completely disappears after 10 weeks of treatment. But the good thing about this model is that when you stop feeding your mice with cuprizone, they actually recover, and we were able to observe this quantitatively using magnetization transfer. This technique doesn't only produce these specific parameters, but also some others, which are the, for example, the most relevant ones, because not measurable in other ways, are the forward exchange rate, RM0B, and the T2 of the macromolecular pool. So I would say that a lot less research is concentrated on the use of these parameters, because it's more difficult to understand what they represent. But a lot of the work that I did while I was in London was on trying to make this technique more reliable and really reduce the scan time so that it could be included in clinical protocols. And this was really the final work that I did when I was there in collaboration with Danny Alexander, who works at UCL. What we did was basically to use mathematical methods to optimize the distribution of the points that I showed you in the previous slide. So it turns out that if you are very clever in the way you place that points, you can actually extract a much better quality fit than if you just use the most obvious set that you can think of. So this is an example of how the quality of the parametric maps can be improved by using, and so these two scans were obtained exactly in the same time on the same volunteer. And I always include this slide in my presentations, which is called Why Pain and MR Physicist, because these were the first quantitative MT maps that the group published in 2003, and after 3 years of working really hard on optimizing the technique, this is what we obtained. So I think that the images really speak for themselves. In September of 2007, I decided that it was time to go and enjoy a little bit of warm weather and moved back to Italy, and in particular I moved to Rome, where I joined the neuroimaging laboratory at the Santa Lucia Foundation, which is an institute for research and hospitalization, and one of the focus of the hospital is really on aging and dementia. So one of the things that I did when I moved over there was to try and apply all these methods to the study of dementia. And we thought that it would have been interesting to apply the magnetization transfer in the study of Alzheimer's disease. And to be completely honest with you, the motivation for doing that was to try and look at the white matter and see whether we could observe some involvement of myelin. We didn't really find any evidence for that, but to our surprise, we found spread changes in another magnetization transfer parameter, which is the exchange rate. So this 3D rendering of the brain show in red the areas where these changes were located. And what captured our attention was the fact that this distribution very strictly resembles the typical distributions of abnormal glucose uptake measured by PET in Alzheimer's disease. So especially in the posterior areas of the brain, these patterns really match each other. So there's a lot of different interpretation of this, but what we came up with was based on the observation that this parameter is known to be correlated with the pH, which measure acidity of tissue. And so we thought that a decrease in the pH could be a sign of metabolic change, and this would match the finding using PET, which would be related to dysfunction of the mitochondria, which are like the power stations of our cells. So these mitochondria are known to play a role in aging, and there's some literature that suggests that they also play a major role in Alzheimer's disease. And while I was in Rome, I became interested in another area that is becoming quite big in neuroscience, which is the concept of brain connectivity. And the reason why this is becoming really popular is because there's a whole school of thought that says that a lot of brain disorders are really related to a problem of disconnection between different areas of the brain. And the people often talk about functional and structural connectivity as two separate concepts. So just to introduce how we can measure connectivity in the brain, I will just briefly explain what functional MRI is. Basically, if you take a volunteer, put them in the scanner and ask them to perform some task or to be subjected to some stimulus that is on for some time and then alternates with periods when it's off, and you take serial images of the brain, then you will observe that in some specific area of the brain there will be some fluctuations of the MR signal that, once that you get rid of the noise, resembles very much the trend in your stimulus presentation. And this is due to the fact that when a specific area of the brain is working hard, there is an increase in the oxygenated blood that is delivered to that part of the brain. And oxygenated blood has different magnetic properties than deoxygenated blood, and therefore we're able to detect this using MRI. So if you cleverly look for all the areas of the brain that follow this pattern, you're going to be able to build a map that shows you which specific parts of the brain are involved with that specific task. So this has been known for quite a while, but what has become apparent more recently is that actually our brain is never really at rest. And so even if you take your participant and ask them just to lie in the scanner without doing anything and take serial images of the brain, then you will still observe some fluctuations in the signal. And what is even more interesting is that this fluctuation in the signal will be concomitant in different areas of the brain, suggesting that there is some kind of connection between areas of the brain that are not necessarily adjacent to each other. So if you look for all the patterns of signal fluctuation and how they relate to each other in different areas of the brain, you can reconstruct maps of what are believed to be networks subserving specific functions. So this technique is known as resting state fMRI for the fact that it reflects the activity of the brain at rest. The mechanism behind it is believed to be some sort of standby status, meaning that our brain is there, it's at rest, but at the same time, these networks are ready to be switched on so that once some activity is requested, this can happen very efficiently. So this image here represents some of the most commonly reported resting state networks, of which the first one on the top, which is the default mode network, is by far the one that has been the most widely investigated. So one of the interesting things of the default mode network is that it's been now shown by different groups that the strength of the correlation between the signal in these different areas of the brain tends to diminish in some conditions such as Alzheimer's disease. And this is one of the studies that we did in Rome using this technique, showing that in the preclinical phase of Alzheimer's disease, you can already measure some changes in the connectivity of the brain long before you can measure atrophy in the same area of the brain. So it's like this functional disconnection precedes the appearance of a macroscopic atrophy. So because of this evidence, we thought that it would be very important to try and investigate whether there is evidence for anatomical or structural disconnection, which would provide also a mechanism for the spread of the disease across the brain. And because the structural connections in the brain are indeed provided by the white matter, it seemed to make sense to think of using diffusion imaging to look at these connections. But even though it seemed an obvious choice to think of diffusion imaging, it wasn't actually so obvious to think of what parameter to use to measure this connection, because sometimes people suggest that diffusion anisotropy is a measure of connectivity. But I'm sure that I can convince you that that's not the case by assuming that the underground lines in London represent white matter fibers. So when we measure diffusion, we actually look at diffusion properties in a cube that represents the image pixel that is much larger than the white matter fibers themselves. So it contains millions of these fibers. And it's a little bit similar to what happens in this situation here. If we were to measure diffusion anisotropy here, we would find that that's very high, and there is a very consistent direction of diffusion. While if we tried to measure it in the West End, because of the presence of fibers running in all directions, the anisotropy would actually be quite low. But I think none of you would try to convince me that northern London is better connected to the rest of the city than is the West End. So something similar could be argued about the brain. So we decided that anisotropy was not going to be the best way to approach this, while because of the properties of diffusion to extract the principal direction of white matter fibers, this has led to the concept of diffusion tractography, which actually aims at reconstructing white matter connections. And so we did this work in collaboration with Jeff Parker in Manchester. Oops. And the idea was to try and reconstruct the white matter connections that run through every little bit of the brain, and then count them to obtain a map of all the possible connections, a quantitative map of all the possible connections that the brain has with other areas of the brain. So it's a sort of visitation map that tells us how much every single bit of the brain is connected with the rest of the brain. And we did indeed apply this to a population of patients with Alzheimer's disease. And what we found was that this index was reduced in several areas, and particular white matter tracts that constitute essential connections for the default mode network, and therefore fits very well with our original hypothesis. So basically, this terminates my lecture, because in the June of 2011, I eventually moved back to the UK again, and I ended up in Brighton, where I now work at the Clinical Imaging Sciences Centre on this campus, and which is really a place that has been a great pleasure to work there since I have arrived, and hopefully it will also lead to a lot of interesting studies, at least of the same entity as the ones that I've presented to you before. But before I finish, I just wanted to have some special thanks to Robert Sowers, who realized the poster for this lecture, which I think all of you will appreciate is really wonderful. Gareth Barker for providing a lot of the pictures of the people that featured in this presentation, some of which maybe these people wouldn't have wanted to see. And then I just want to thank Marco Bozzali, who not only has been a collaborator in a lot of the studies that I've presented tonight, but also my partner in life. My sister and my mom, who have always supported me during my career, despite my jumping up and down different countries and all that. But I also wanted to dedicate this lecture to my father, who was an academic himself and has certainly set a high example, not easy to reach, but hopefully would have been proud if he had been here tonight. So thank you very much.